A review of the mock exam paper, question by question. If you answer the question correctly, then fast forward on to the next question and make notes on any question that you failed to answer fully correctly. Question number one, question is about respiratory pigments in muscle cells. You should be thinking about the one respiratory pigment you know something about, the protein, hemoglobin, and looking at the structure on screen. The unique feature about it is that it contains iron within the molecule of the protein, which is a prosthetic group. So prosthetic group is the answer A. Question number two. A student has used a colorimeter, and if you look at the graph, we've got on the two axes, on the bottom x-axis, the concentration of the sugar. Now, the unusual thing is that fully concentrated sugar with a number of 1 gives you an absorption reading on the colorimeter of 0. Whereas if you've got no sugar at all, 0 concentration of sugar, that gives you an absorption reading of 100%. So clearly the lower the number, the more concentrated the sugar is. The question is, which of the rows, A, B, C, or D, shows the correct sequence of sugars from highest concentration to lowest? So remembering a very high concentration has an absorption of 0. Looking at P, Q, R, and S, the lowest number is 40. So that will be the most concentrated, followed by P, which is 60. The next one is R with 70, and the weakest sugar is S, where 100%. So the correct answer is QPRS. Next question, talking about the polymer collagen, and it's saying that the monomer from which collagen is made, you need to name it. Collagen is a protein. All proteins are made from amino acids. So the first one is amino acid. It then says, how are they joined? Now that's not very specific. You could name the bond or the process by which the two amino acids are joined. So if you said peptide bond, you get a mark. Or if you actually said it was a condensation reaction whereby water is produced, you would also get a mark, giving you a total of 3. We've now got a food test. Benedict's is the chemical used for testing for proteins, for sugars, and Biuret is the chemical for testing for proteins. Benedict's solution starts off at blue, and if it reacts with a sugar, it will turn brick red. And the test it's showing is non-reducing sugars, it turns brick red. Which of these is a non-reducing sugar? The answer is C, which is sucrose. A student mixes an unknown substance with water and ethanol. Now that should remind you the standard test for a fat, a lipid, is adding first of all to ethanol and then adding to water, and you should get a cloudy suspension if they're present. So the test is for lipids. A vet is concerned that a llama is unwell. So a llama being an unusual animal, but the vet suspects that hemoglobin may be in the urine of the llama. Again, hemoglobin is a protein. So if we do the protein food test, if it's positive, it means there is hemoglobin in the urine. So you just needed to explain that you carry out the Biuret test, which is made up of sodium hydroxide and copper sulfate. You did not need to state that, but you did need to state that you add Biuret to the urine, and then you need to state that if it's positive, it will go change color. So therefore, you need to state the color change, which for Biuret starts off at blue and will go purple, confirming that there is protein in the urine. Question number 7, which row A to D is correct when describing the 4 proteins, collagen, insulin, elastin, and hemoglobin? And the correct answer is row B, because collagen is a fibrous protein but doesn't stretch. We find collagen at the end of muscles attaching it to bones in tendons. Insulin is a globular protein and the shape never changes. Whereas if you look at the answer at the end, hemoglobin is also a globular protein, but it changes shape depending on how much oxygen is attached. Elastin, in the name elastic, is a fibrous protein, but it recoils, it regains its shape when you pull it. So B is the correct answer. The statement here is that here is a standard molecule of glucose, and it tells you it's beta glucose. And the first part of the question is, can you label X, Y, and Z? X is carbon, Y is oxygen, and Z, because we've got the hydrogen above, will be OH. And you didn't have to name the molecules, you just had to put the chemical symbols. Describe how the structure from the molecule of beta glucose would be different if it represented a beta glucose. So if an alpha glucose was changed into a beta, how would it be different? You should point out that it is carbon 1 where the hydrogen and the OH are reversed. So a simple diagram would have been sufficient to gain the 2 marks. Third part of the question. 2 glucose molecules can be joined together to form a disaccharide. What is the precise name of the bond that forms between 2 glucose molecules, and what would that particular disaccharide be? So you have to put that the bond is the alpha 1,4 glycosidic bond. You cannot put down when you've got 2 glucoses that it's just the 1,4 glycosidic bond. To get the mark, you have to put alpha 1,4. And secondly, the sugar that's formed when glucose joins with glucose to form a disaccharide is maltose. Glucose, glycogen, and amylase are carbohydrates. Now, this question is a 7-mark question. It gives you some basic information. Glycogen and amylose are used for storage. Glycogen's in animals, amylose is in plants. Describe how the structure of glucose allows it to perform its function, and explain the advantages to animals of using glycogen as an energy store. So first of all, you need to focus on glycogen, not on amylose. That's been put in as a bit of a red herring. So glycogen is what they want you to describe the structure, and then you need to talk about how it gives an advantage to the animal as an energy store. And here is a whole range of bits of information which would have scored you maximum marks if you had 7 of these various answers. So running from the top, glycogen is made up of alpha glucose. It can be broken down by hydrolysis. You must use the keywords. Glycogen is a polysaccharide, very long-chained molecule. Glycogen is insoluble in water. What's the consequence of that? It does not have a water potential, so it will not be affected by osmosis. Why does it have the structure it does? Well, it enables a large amount of glucose energy molecules to be contained within a fairly small volume. It's also got branches, side branches, and they are the 1,6 branches. And therefore the enzymes can actually attach wherever there is a 3 glucose. So the 1,6 bonds give lots of sticking out glucoses. So you can have multiple sites of enzymes breaking the glycogen down. Why do we need multiple sites? So that we can get lots of glucose quickly. And why do we need that? That leads on to the final part. Animals have a large metabolic rate, a high metabolic rate, because they move, they hunt, or they have to run from something that's hunting them. So they have a high metabolic rate, therefore they need a lot of energy, and that's the reason why glycogen is a perfect molecule for them. An even higher scoring question is about red blood cells, erythrocytes. Erythrocytes contain hemoglobin, which is a globular protein. Blood vessels contain collagen, which is a fibrous protein. Describe the differences between a globular and a fibrous protein using hemoglobin and collagen as your example. So you may know about another named molecule other than hemoglobin that is a globular protein, such as an enzyme. You're not required to discuss them. You've got to focus your answer only on these 2 molecules. So to score the full 8 marks, you should start off comparing and contrasting one of them. So on the left-hand side, we have all the features that you could have raised regarding the globular protein hemoglobin. First of all, like all globular proteins, it has a ball-shaped spherical shape. It has hydrophilic molecules on the inside and hydrophilic, water-loving on the outside and hydrophobic on the inside. It forms hydrogen bonds with water. It's soluble, and hemoglobin is a good example. Moving on to what hemoglobin does, it is a transport molecule for transporting oxygen, and it also can transport carbon dioxide. Hemoglobin, as we know from the very first question, contains iron, which is a prosthetic group. Again, remember the examiner is looking for you to use key words to get these marks. So use the word prosthetic group because it contains iron, which is a mineral. And it has a unique tertiary and a quaternary structure as well. Moving across to the fibrous molecule, which is collagen. First of all, talking generalizations about fibrous molecules. They're long, they're strong, and they have a structural role. Collagen, if you remember, has a high proportion of the amino acid glycine, and so the chains can lie very, very close together. There are cross-links between the fibers of collagen, making the molecule very strong. And we can then finally state where collagen is found. It is found in ligaments, tendons, attaching muscles to bones. Question 10. The R group in the amino acid aniline is CH3. Now, you should remember and be able to recreate the basic structure of an amino acid. They all have the same basic structure. The R group is what changes, but here they're telling you that the simplest amino acid, aniline, the R group is CH3, and it wants you to describe the structure. What better way than to draw the molecule? So if you simply draw the molecule, the carbon in the middle, COOH on the right, NH2 on the left, hydrogen above, and instead of putting R underneath, you put CH3, you would have scored 4 marks. Sticking with the plant proteins, amylose and cellulose, it wants you to compare and contrast by putting down at least 3 features where they are either similar or different. So the answers that you could have had, any 3 of these on the left and any 3 on the right, is telling you that amylose is coiled and cellulose is not coiled. Amylose is made up of alpha glucose, whereas cellulose is made up of beta glucose. The alpha glucose is connected by the alpha 1,4 glycosidic bonds, and whereas the cellulose is by the beta 1,4 glycosidic bonds. The monomers, the amino acids making up the structure of amylose, are all in the same orientation, whereas the one for cellulose, if you look at the picture, we have hydrogen above and then hydrogen below on alternative molecules. It is like a thumb pointing up and a thumb pointing down. They're rotated by 180 degrees. The amylose are not fibrous, they're not structural, whereas cellulose is fibrous. And we have hydrogen bonds on both of them, but the big difference is the hydrogen bonds are within the molecule itself, whereas on cellulose you have hydrogen bonds joining cross members to make the structure very, very strong. There was a supplementary question, which is what properties of cellulose make it suitable for forming cell walls? Well, it has to be strong, so using the word strong would have got you 1 mark. And secondly, it is insoluble in water, gives you the 2nd mark. Question 13. Proteins are important biological molecules. Hydrogen bonds form between water molecules. Describe the formation of hydrogen bonds between 2 water molecules and explain why water can form these bonds. So basically, a picture as shown below is an ideal way. You must label it delta plus and delta minus. You must show the delta plus on the hydrogen and the -1 on the oxygen, and that would have been sufficient to gain all the marks for this question. Water can act as a solvent. Why is the ability of water to act as a solvent important? Now, don't just simply ramble on about different properties of water, such as its ability to act transporting water up a plant through adhesion and cohesion. That has nothing to do with the word solvent. Solvent is something that dissolves other molecules. So water is involved in metabolic reactions. Why? Because it can dissolve various minerals, such as sodium chloride. Always name an example. Its function as a solvent is to transport these molecules around, and you could either talk about it being transporting plants through the tubes called the xylem, or you could say it is transporting in the blood vessels of animals. Water also, being a solvent, can dilute molecules. So therefore, that's another property of it being a solvent. Any three would have scored you 4 marks. A nice easy question here. We have a glycerol molecule and a fatty acid molecule. And normally you have 3 fatty acids joined to the OHs on the right-hand side to form a triglyceride. The question, however, asks you in the space below, draw a monoglyceride. So you only use one of the fatty acids and you leave the other 2 OHs alone. So diagram is what was required. And here you can see I've highlighted the hydrogen of the glycerol and the OH of the fatty acid molecule. That is water. If you remove it, it's a condensation reaction. And what are you left with? You're left with an oxygen from the glycerol molecule, which then joins on to the carbon of the fatty acid. And then you just had to draw the rest of the carbons and the hydrogens to get 2 marks. Question 14. We have a graph. We have enzyme activity showing 100% on the y-axis, and it drops down dramatically for 60-degree temperature. It drops down but less rapidly for the 50, and it is not dropping down for 30. The question is, explain the shape of the curves at 50 and 60. So you should not really be mentioning 30 degrees. What we can see is a variety of factors about enzymes and temperature. So first of all, 50 and 60, both of them are being denatured, which is why the percentage of activity is dropping. 60 degrees, the rate of drop is quicker. It's nearly all deactivated by 30 minutes, whereas at 180 minutes, the 50-degree enzyme is still working, even though it's much reduced. Why is this happening? At high temperatures, the hydrogen bonds break down. And what effect would breaking the hydrogen bonds be on a protein? It will alter the active site, and therefore it loses its shape, and therefore it's unable to carry out its enzymic reactions. A new question, but again related to enzymes. Some proteases, they're enzymes that break down proteins, are secreted as extracellular enzymes by bacteria. So rather than bacteria taking in food, they pour the enzymes on top of their food. Select one advantage of a bacterium secreting enzymes into its natural environment. Why would it be doing this? Well, clearly to feed. To digest proteins, and once it's digested the proteins with the enzyme, it will reabsorb the amino acids. It could be, it's not food, it could be a toxin, so it could be pouring its enzyme on top of a protein toxin to kill it, to destroy it. Mammals have extracellular proteases, but they also have some that are bound to the membrane. Describe the action of these membrane-bound dipeptides. So the same thing, it's asking you, how does the enzyme work? The enzymes work in the same way by doing hydrolysis to release amino acids. The dipeptides cannot pass into the cell because they're attached to the cell membrane, and that stops the enzyme from entering the cell and digesting its own contents. So simply being attached to the membrane means that it is being held in place. And again, it can be reused again and again because it's not being secreted like the one on the bacterial example in the previous question. We have a diagram of a structure drawn by a student, and the first part of the question is suggest 2 ways the student could improve the quality of her scientific drawing of this fish gill. So there's more than 2, but the 3 that I've highlighted are, first of all, we have shading. You're not allowed to shade on the biological diagram. Moving up, we have broken lines. And thirdly, the label, the hair and the cuticle are crossing. You never have labels that are crossing. They always have to be horizontal and not touching each other. Scientists are involved with the treatment of a bladder infection caused by a bacteria, and they're saying that they've developed a new antibiotic. How does it work? It inhibits the bacterial ATP synthase enzyme. So ATP synthase tells you that it synthesizes, which means it makes ATP. So automatically now, what do we need to make ATP? We need ADP and phosphate. They're the 2 things we need. So which of these are correct? You can ignore the ATP on the left-hand side, which is the first 2, but then we've got the 2 below. Water is not involved. You only need ADP and phosphate, and it will form ATP and it will produce water. So the answer is ADP plus phosphate goes to form ATP plus water. The new antibiotic is safe to use in humans, and it does not inhibit the ATP synthase found in human cells. So why is this chemical affecting bacteria ATP synthase but not humans? Clearly, the structure must be different. So when you give the answer, you can say that the structure is different, use keywords, the tertiary structure is different, it has a different shape, is good enough to score the marks. Final question is on enzymes, but enzymes associated with the manufacture of DNA. So the 2 enzymes involved is the one that undoes the double helix, which is helicase, or more correctly, DNA helicase, which causes the hydrogen bonds to break down and the 2 strands to separate. And then when you add new nucleotides to each of those 3 DNA chains, you need to stick them with DNA polymerase. So they're the name of the 2 enzymes.